Hello, I'm Claire Reimers, President-elect of the Ocean Sciences section of AGU. This year's Rachel Carson Lecture will be given by Dr. Drew Harvell, who exemplifies Rachel Carson's work through her own brand of relevant and cutting-edge ocean science. Dr. Harvell is Professor of Ecology and Evolutionary Biology at Cornell University and the curator of Cornell's Blaschka Glass Marine Invertebrate Collection. She is also on the board of the University of Washington's Friday Harbor Marine Labs. Her research focuses on the health and sustainability of marine ecosystems. Her fieldwork has taken her from the coral reefs of Mexico, Indonesia, and Hawaii to the seagrass beds of the Pacific Northwest. And it's resulted in over 170 academic articles in journals such as Science, Nature, and Science Advances. She is a fellow of the Ecological Society of America and the Atkinson Center for a Sustainable Future. Dr. Harvell was awarded the 2020 Cornell SUNY Chancellor's Award for Excellence in Research, the 2019 Seattle Aquarium Conservation Research Award, and the 2019 Northwest Yachting Magazine Outstanding Environmental Leadership Award. Her first book, A Sea of Glass, was published in 2016, and it was named for the National Outdoor Book Award and given honorable mention for the Rachel Carson Environmental Book Award. Her newest book, Ocean Outbreak, is also award-winning and highly timely as it details increased infectious epidemics in the ocean and solutions that benefit people and biodiversity. During our global human pandemic, she reports she's been able to do field work in the San Juan Islands and has acquired a new pandemic puppy, for which I'm personally quite jealous. Dr. Harvell is passionate about communicating science. In addition to her award-winning books, she's written for the New York Times, CNN, and The Hill. So her lecture today is entitled Oceanic Pandemics from Foundation to Keystone Species, and there will be a live question and answer session immediately following the lecture.
Good morning. Thank you to Claire and the Rachel Carson Committee for this great honor. Today, I'll bring to life some of the pandemics raging in our oceans, recounting work done by my colleagues and some of our current work with marine disease. While I will emphasize the dangers faced by the role of climate warming in facilitating these infectious diseases, I don't want to lose sight of the fact that these frontiers and opportunities of using our science and innovation to get ahead of this juggernaut are immense. So while an epidemic as wide-ranging and destructive as COVID is new to our modern society, large-scale destructive disease outbreaks are an age-old process in our oceans. These outbreaks are rapidly accelerating now with climate change, and infectious disease pandemics of the last 50 years have reshaped coral reefs, seagrass beds, our intertidal, our Pacific seascapes, aquaculture practices on salmon and shrimp farms. These disease outbreaks are transformative when they attack foundation species like seagrasses or keystone predators like seastars. It's not just an honor to give a lecture named for Rachel Carson, but it's an inspiration since it was a chance to review anew her books. I found at every step she'd thought creatively about many of the biotic processes that are important today. While I think most of us here associate often with the take-home message of Silent Spring, that industrial synthetic chemicals like DDT are killing us and our biota. And we definitely celebrate her victory in getting new life-giving legislation passed. I was inspired to see so many ideas that are germane to the issues I face with marine disease outbreak, and I'll bring up a few of those as we go along. Here's one. Nature has introduced great variety into the landscape, but man has displayed a passion for simplifying it. Thus, he undoes the built-in checks and balances by which nature holds the species within bounds, and that certainly resonated with me. And so while Rachel Carson warned that toxic chemicals were the priority of her time, I'd like to warn that climate change can act directly and through biotic interactions to unravel ocean ecosystems in ways we might not have been really thinking about. And I've had a chance to talk about this a lot, or think about this a lot, not talk so much about it. We've just finished a 6-year research coordination network that involved over 50 scientists. And this was our main focus, was ocean infectious disease and climate change. And as a result, we've got a bunch of papers out, not just by me. Colleen Burge led a paper on warming climate and disease risk, Jeff Maynard on forecasting warming and disease, Kevin Lafferty on economic impacts to fisheries, Allison Tracy on baselines. And I did write a book, and this was intended for a wider audience as part of a broader impact to try to reach out and send some messages to others. So I'll talk about some of those today. All of the work that we do within this group, but also within my lab group on the ecology of host-pathogen interactions, also serves as a way to look at sentinels of change in ecological interactions. And we pretty much always study our hosts and pathogens in the context of a changing environment. And just to remind you, how do we define a pandemic? And it was interesting that World Health Organization kind of struggled with this quite a bit at the time. The definition I've seen is it's an epidemic occurring worldwide or over a very wide area, crossing international boundaries. And we normally emphasize the crossing international boundaries. It doesn't have to be worldwide. But I just want to point out that 3 of the outbreaks that I'll talk about today and that we've worked on would be classified as pandemics in the sense that they've crossed international boundaries. And these are diseases of corals, seagrasses, or some of our keystone species like seastars. And so I'll talk in general about some of the work that we've done and others on infectious epidemics in the ocean increasing with warming. I'll focus most on outbreaks in eelgrass meadows from San Diego to Alaska, because this is our current project and the one I think might be of most interest to this group. But I want to end by talking about some of the things we can do better, ecosystem services to improve ocean health. I'd like to start with this paper by Jeff Maynard and other members of our research coordination network, because it very nicely summarizes temperature-sensitive diseases of marine taxa. So these are diseases that are below their thermal optimum and that will increase under warming conditions. And so you can kind of see the range of taxa that are represented here. And the very clever thing that Jeff and colleagues did in this paper is to actually attempt to forecast, because some of these are so temperature sensitive, and he picked lobster shell disease to forecast how these outbreaks would change in Maine over time, of course, where the economic impacts would be great. And so these are some of the methods that are important to develop under this scenario. The most central number, and it's one that actually I think we've all come to know, is the R0, the basic reproductive number of an infection. What it means is the average number of new cases generated from each infected individual over the course of an infection. Now, as you know, it's an average, so it's a population average of these. And these are just some of the R0s for situations you may know well. Measles is an extremely infectious disease with an R0 of 12 to 18. So 1 is the important number, because an R0 of 1 means it will just barely increase through time. Anything less than 1 and the disease won't propagate. So you can see that Ebola while it was a horrifying disease outbreak, had a relatively low infectious potential, just over 1.5, and yet it spread very widely. Similarly, influenza is an R naught of 1. And of course, you've seen some of the recordings of coronavirus at 2 to 4. And everybody understands, I think right now in this moment, if you think about an R naught for coronavirus, it's totally dependent on the context. It depends on the season. It depends on the town you live in. And so all of these R naughts are global averages. I don't know yet. One of our plans is to figure out the R naught for our eelgrass wasting disease. I'm guessing at about 7. And I'll tell you about some of how infectious it is as we go along. I think this is a useful way to think about climate warming disease impacts. This is a plot of temperature on our y-axis through time, and then the R naught. And so this blue line here shows sort of the old blue oceans and a disease. And you can see this R naught is plotted here in blue, and it increases through time. It's only for a short period in the summer that it actually crosses this severe disease period where maybe the R naught is 2 or 3. And then it declines in the winter again. However, with warming, this whole relationship can shift upwards. And thus you might have an R naught that's above this pink line of severe disease for most of the year. And that's the situation we're finding in some of our ecosystems with coral and eelgrass diseases. So one of the projects of our research coordination network was to figure out baselines of marine disease. There are no baselines for marine disease. And so what Allison Tracy, then a PhD student in my lab and now a postdoc at the Smithsonian, did is she and her colleagues worked with 40 years of disease records. And what's plotted here, this is the sum of normalized disease reports for a whole, like all of our major marine taxa, from sea urchins to fish. And so for this number here for fish, what this means at a little over 4% is this is the percentage of disease reports normalized by all the literature on fish. And so while that seems very small, that's over 40 years, and it varies a lot through time. We focused on coral because that's one of the disease outbreaks that's very related with temperature. And what you can see here, if you plot, again, this is our 40-year period from 1970, and you plot the percent of reefs temperature stressed and the disease reports per taxon. The black is the disease reports per taxon. And this is one of the cases that increased significantly through time. You can see the big increase in our recent years. And you can see this is correlated significantly with warming. So we don't know for all of these how these baselines have changed through time or what the drivers are, but for at least one of them, we can link it with warming oceans. And this is just one example of this new tissue loss disease that's now spread Caribbean wide and is causing enormous damage because it's a very rapidly propagating infectious disease that has a wide host range. I want to briefly talk about some of the work we've done with seastar outbreaks. And the reason to call this one out is it's been designated as the largest marine wildlife epizootic in the ocean. And it's considered to be so large because of the wide host range. Over 20 species, well over 20 were affected, and well over a very wide geographic range from Mexico to Alaska. It started in 2013, and unfortunately it continues on the West Coast. And it's affected ecologically important keystone and predator species. These are just some of the news reports that came out as we were doing this work. And rather than telling you about the work that we've done, I'm going to play this video abstract about a paper that we wrote a year ago talking about the extreme impact to the most susceptible species in this outbreak.
There's an epidemic in the ocean. Since 2013, a viral disease has been turning sea stars in the Northeast Pacific into melted piles of goo. Of the 20 or so species of sea stars affected by the virus, one of the hardest hit were sunflower stars. Until now, we haven't known just how bad the decline was, but new research has begun to reveal the longer-term continental scale impact of the epidemic on certain species. Scientists in the US are now suggesting we formally list the once common sunflower star as an endangered species. Trained citizen science divers from California to Alaska counted sunflower stars on over 11,000 dives, while scientific divers from the Hackeye Institute carried out more detailed surveys on the BC Central Coast. When they looked at all the data, scientists noticed something in common where they saw outbreaks of the virus: anomalously warm water. We still don't know how these warm water anomalies and the virus interact, but researchers say these warmer than normal water temperatures were related to dramatic sea star declines. While divers can patrol waters near the surface, we didn't know whether sunflower sea stars might have found refuge at deeper depths. But thousands of NOAA bottom trawl surveys have revealed that when it comes to sunflower stars, the disease didn't stay in the shallows. For example, data from Washington State shows a crash in populations in both shallow nearshore and deep offshore environments after the epidemic began in 2013. Data from other areas on the coast are similar, with population declines of as much as 80 to 100% in areas across the 3,000 kilometers from Alaska to California.
So I'm going to stop that and move on with my talk. I just want to really kind of emphasize this because it's a case where we've had a large ecological impact from an unexpectedly broad, fast-moving infectious disease. And we also relied heavily on citizen science diver data. This is some of the Reef Check California data, and I want to point out this goes back to 2007, so that we know the baseline in this case of how this population was doing through time. And you can see the big crash in 2014. And what I want to emphasize here is the trophic cascade associated with the decline of this pivotal species and the rise of its prey, sea urchins. So throughout California, the species is essentially gone now, and it used to be very common. Urchins have taken over and they've mowed down kelp beds, and so large consequences. And as a result, there's interest in a recovery plan. So just to briefly update what I've talked about for the sea star epidemic, this species Pycnopodia is now rare. It's imperiled and still dying in many parts of its range where it exists. Other species are still experiencing mortality events and are still quite rare at our San Juan Island sites compared to what they were at the start. On the other hand, there's also interesting, exciting evidence that some species have a degree of resistance, such as the ochre star. So there's a lot to be learned about that, and a high degree of uncertainty about the causative virus. So I want to just say that there's underway a Pycnopodia, the Sunflower Star Recovery Plan, and it's being launched by the Seattle Aquarium, the Nature Conservancy Canada, and about 30 different scientists. It includes things like captive breeding at Friday Harbor Labs, and Sarah Graveham and Sarah Hamilton actually have prepared a listing as imperiled with the IUCN. And so again, just because of the wide range and the fact that this is an endemic species and ecologically important. And so just to go back to that, there's a 90% global decline and 97 to 98% if you just count the continental US West Coast. So that's my 2 cents worth about how important these outbreaks can be. You can't actually answer a polling the way we have this set up, but I want you to think about it. Take a minute here. So why is the sunflower star uniquely imperiled by a disease outbreak? And if we could poll, I would give you these options. I'd say, is it that its habitat puts it more at risk? Does its food put it more at risk? Or is it the most susceptible to a multi-host pathogen? And I'll give you a minute to think about it. But I'm thinking most of you, I can hear you. I think you said number 3. It's the most susceptible to a multi-host pathogen. And of course, we know that multi-host pathogens have caused other problems with biodiversity. The Hawaiian birds are a perfect example with avian malaria and massive extinctions. I want to turn now to a different habitat. I'd like to talk about eelgrass meadows because this is such a vital marine habitat with important services. And those services include things like essential herring spawning, juvenile salmon habitat, blue carbon storage, and filtration services. And I'm actually going to come back to the filtration services at the end because of its potential to be important for diseases. However, our eelgrass meadows are affected by a protozoan that causes these kinds of lesions and this kind of black melanization response by the plant. And the kinds of questions that we're asking in terms of whether this is a keystone pathogen is, first, I want to convince you that we're doing a pretty good job. No, we're doing an excellent job diagnosing it. I want to take you through some of those steps. I want to talk about how prevalent it is in nature, how it's transmitted, and how we're scaling up with some new tools to get wider ranging estimates of these diseases that extend across multiple latitudes. Here's what it looks like. These are the spindle-shaped cells of the Labyrinthula zosteri. It's a protozoan, and it runs in these little mucus networks it lays down inside the eelgrass tissue. So this is the histology showing the little cells. In terms of our diagnostics, we've worked really hard over the last decade. One, to be able to culture this consistently, to be able to identify it in histological sections, and now with molecular methods. I'll take you through a little of that because I want you to be confident in our diagnosis of this disease. So here's what it looks like. So these are a bunch of, this is all one plant here. Oh, sorry, one plant here, one plant here. And you can see sort of the different forms that this disease can take. And we needed to be able to say, is this the disease? And is this not the disease? And I think we can answer that question. These are the results of a qPCR test validation that Morgan Eisenlord and Colleen Burge have developed. And so, for example, these solid lesions like this have very high cell counts, and the phenology experiment I'm about to show you reveals that these are early infections with high cell counts. And as the infections progress and become more necrotic, the cell counts drop down. Control blades are consistently clean with no or very, very low cell counts, which of course is important. The other thing that's important about this disease is that we can culture it. And so this is an experiment that Morgan and Phoebe Dawkins ran. And you can see these controls are, this is the control, and this is the experimentally inoculated blades up to 20 days. And in this experiment, the controls remained largely asymptomatic, and the disease severity on these blades was quite high, starting at 2 days. This is what the plants look like. So these, again, are inoculated with pure culture in the lab, developed lesions through time. These are the qPCR readings from that experiment, again, to kind of put together the whole story of an inoculation and the phenology through time. And you can see by 2 days, pretty high, almost 8,000 copies per milligram of tissue, up to a high average with a lot of variation through time. And so with that as a base, we can turn to the field and try to understand what are really pressing knowledge gaps in nature, such as how does this thing transmit? And so we knew that if we put sentinel blades inside eelgrass beds, that they would probably get infected, because we knew that contact was one mechanism of transmission. What we did not know was if our sentinel plants outplanted 100 meters and 300 meters would also get infected. We actually did not think the 300-meter plants would get infected because we were assuming these beds were our source. The results were pretty exciting, and they've kind of opened up a new realm in our study, which is that inside the bed, roughly 68% of these sentinel plants that were asymptomatic when we put them out at the beginning of 2 weeks were infected. Our near bed were also infected at a statistically indistinguishable level from the inside the bed, and our 300 or actually 400 meters from the bed plants. And so this is clear evidence of waterborne transmission of this disease and the highly infectious nature. Also now working with field populations. These are time series studies we've done over the last 7 years. And of course, we've had some warm periods, and it allows us to test the effects of warming in nature. We found extremely high levels of over 50% in the last 3 to 4 years. So it's been increasing through time to quite destructive levels. The interesting thing in this this paper that Maya Groner is the lead on, that's in review, is that we were monitoring plants through a big heat wave. This is the winter temperature anomaly in 2015 and 16 in our San Juan Island waters. Here's the West Coast. Here's where San Juan Island is. I'm here right now. And then this is the spring temperature anomaly. And so in the summer following these warm anomalies, our plants had higher levels of disease. And so this is the evidence that warming can be contributing to the outbreaks that we're seeing. And in this modeling that Maya did, let's just look at 2015 because there's a lot of plots here. And what you can see is this, for example, would be a comparison between beds for one winter for the effect of the spring warming. And you can see that as there's warmer temperatures at some sites in May and June, they have a higher level of disease. These multiple lines reflect sites that have different degrees of winter warming. And so if there's no cold days in the winter, no winter days below 8 degrees, there's a very high predicted disease risk. If there's a lot of cold days at some sites, they're much less likely to be as sick in the summer. And so this is sort of the beginning of ferreting out these seasonal and spatial patterns due to warming. To do that, we wanted to scale up to much wider studies. And so this is the beginning of a project that spans the entire West Coast from San Diego to Alaska. And to do that and get the diagnostics right, we needed to have an objective tool for analyzing disease. And so in order to be able to assay several thousand plants at a time, we developed this artificial intelligence tool. And I'm not going to do this justice at all. This is actually a really complicated machine language learning image segmentation algorithm developed by Brendan Rapazo and Carla Gomes that took years of training to be able to properly identify and quantify to the 2 to 3% level relative to a trained human. And so with that in hand, we can turn to this new project. This is an NSF grant led by Emmett Duffy and a whole bunch of wonderful partners so that we can assay the health of these plants from San Diego all the way to Alaska. And so the ELISA artificial intelligence algorithm can basically run several thousand of these in a few minutes. And the result, each of these regions, I'm only showing you the mean, but in each of these regions, there's 5 subsites and 8 transects per subsite. So there's a lot of data behind this. What you can see, it's not just me that says Washington State has a problem. We have an average of almost 75% of our blades surveyed in July of 2019 had some sign of disease. You can see that Alaska is also quite high, over 50%, and Bodega Bay quite high. And this study is also incorporating the use of drones to monitor the aerial extent of our beds through time. So this is actually showing us the geographic extent. We also know that beds in Europe are affected by this disease. And finally, the initial data from 2019, and thanks to Lillian Aoki for analyzing all of this data. She's the postdoc on the project. And once again, she finds a link between temperature anomaly and disease prevalence across this wide geographic range. So here's the Washington and the Alaska sites. Here's the Oregon site, which had very few infections and actually a negative temperature anomaly that year. There's a lot of challenges ahead for us. We're particularly focusing now also on the strain variation. We find a lot of, there are different strains that cause these lesions, and they vary in virulence. Some of them cause really rapidly destructive lesions, others do almost nothing. And then we're finding strains that vary a lot that are posing new mysteries for us. For example, this lesion looks very different because of the massive plant response. And when we look at the histology, we see this large melanization response. So we're working to sort out this variation. So I'd like to summarize what I've told you about our eelgrass wasting disease in the context of trying to talk more broadly about these issues of how to study an infectious disease in the ocean. So I'm not going to, again, technically poll you, but I'd like you to think about 4 of my main take-homes and think about which one is of the most interest to you. Maybe you can ask me questions about it later. We've shown that qPCR and culture allow inoculation experiments to work out the phenology of disease. Our artificial intelligence machine language learning lesion recognition allows continental scale. Field and lab infections increased with warming temperatures. I didn't talk about the lab, but we've done that. And disease has waterborne transmission. So now what I'd like to do is turn to what I think of as part 3 of my talk, and I'm calling this ecosystem services per pathogen reduction. And this is my attempt to say something more hopeful and to provide some new ideas for future work. What we found, and this was work that now Professor Jolie Lam, then postdoc in my lab, led in Indonesia, and basically showing that pathogenic bacteria can be reduced by approximately 50% in the Indonesian seagrass beds we were studying. And this was published in this paper in 2017. And of course, I'm going to comment a little bit more about a wider quote from Rachel Carson, but this work reminds me of her comment that man, far from being the overlord of all creation, is himself part of nature. And I hope you'll see why. Here's Baringlumpur Island, where this work started, because we all got sick and then went back to sequence and figure out what was in the water, which was a lot of raw sewage and bacterial pathogens. And it's located here in Sulawesi. This was our study site 3. And so what we observed in our initial sampling was that where they were right adjacent to seagrasses, but seagrasses absent, there were high levels of Enterococcus. But where the seagrass was present, there were very low levels of the Enterococcus, which is an indicator bacterium. These are toxic levels, polluted levels for any water. And then more detailed work showed that these numbers were extremely high close to land, where the sewage was being input, dropped off rapidly, but twice as fast in the green line, which is the seagrass beds, and remained low to the reef. And so I'm not going to show you all the data because it's been published, but there's now, Jolie has complete bacterial sequencing, which is in that paper. And so you can see that for individual pathogenic bacteria, this drop off. What I want to turn to, though, is the insight that human health and environmental health can be linked. And in Jolie's survey, she showed that coral disease is also lower in the seagrass beds. And here's black band disease at half the levels inside the seagrass relative to outside. Whoops. And white syndromes, even more than a half, 50% reduction inside the seagrass relative to outside. So we don't want to say this is the final word on this. It really shows a new direction, I think. And to really kind of drive this home, we've started the work in Seattle's waters and Puget Sound to show this isn't just an issue in Indonesia. And King County, where Seattle is located, has been fined for sewer overflow violations. And these are some of the bacteria that Jolie's team sequenced in our study. What we did here is we worked with the Mussel Watch folks to analyze the mussels that were outplanted in different regions in Seattle. And then we actually put our own mussels inside and outside seagrass beds to use the mussels to integrate bacteria. Here's just a piece of the research from this. This is not published yet, so this is kind of hot off the press. In more urbanized areas, seagrass meadows may reduce human pathogens in mussel gills. So here's the percentage of human pathogens where there's no seagrass and where there's seagrass in these sites that are highly urbanized. Jolie and Evan Fiorenza and Phoebe have also followed up by modeling this as distance to drainage outlet, so the potential human pathogens. What you can see here is, again, the relationship is different if the seagrass is absent than if the seagrass is present. Again, suggesting that seagrasses can buffer some of these bacterial pathogens, which of course is not really a surprising result given the oxygenation provided in seagrass meadows, the degree of filtration by resident invertebrates and other processes. And here's one of those other processes. This is a paper led by Maya Groner and colleagues. And what they showed is that oyster filtration can reduce the severity of eelgrass wasting disease. So here we're asking whether some of the critters that live in an eelgrass bed can reduce the danger of disease for the eelgrass. And so this was run over multiple PCO2 levels, but I don't want to talk about the ocean acidification. I just want to talk about the fact that when oysters were added to the eelgrass, when they were co-cultured, the levels of eelgrass shoot severity were significantly lower at all levels of PCO2. So again, showing the potential for a biotic agent to help. And so in my efforts to try to get a broader message out, and actually when I was very frustrated by how slowly we were responding to COVID in March and April, I was able to get this op-ed in the New York Times called How Starfish, Snails, and Salmon Fight Pandemics. I did not actually write that title, but what I did say was, now the world is seeing the deadly path cut by a terrestrial pandemic spread by a new coronavirus that has killed tens of thousands of people. Of course, now it's over 1 million. If anything good is to emerge from this, it will be in the quest to better understand pathogens and their host, to find nature's best defenses and apply these findings to engineer a safer world. I do believe in that. And I really am impressed by the fact that Rachel Carson said, here and there, awareness is growing that man, far from being the overlord of all creation, is himself part of nature, subject to the same cosmic forces that control all other life. Man's future welfare and probably even his survival depend upon his learning to live in harmony rather than in combat with these forces. And I do think of using ecosystem services to deal with an infectious disease threat is pretty important, and understanding these natural processes in nature. And so 2 of the take-homes from this book that came out last year are infectious disease outbreaks of marine biota are fueled by warming, threatening marine biodiversity, and our food from the sea. So even if you don't care about my invertebrate corals, sea stars, and abalone, salmon are something we eat. And then I talk a lot about how natural marine ecosystems have special powers to reduce pathogen load and indeed huge knowledge gaps. So there's some very exciting science. And so I suggest we put more effort into suggesting host pathogen interactions in nature, and especially these natural solutions to epidemic control. And in that context, I do think the oceans have particular value for expanding our options due to the diversity of pathogens we have. So, of course, I'm talking to an impressive group of ocean scientists and maybe broader group of scientists. And everybody knows that so much of the work that goes on in our labs is done by really talented students, colleagues, and coworkers. And so I really want to highlight these amazing people. I love this picture here because this was our our eelgrass team quite a few years ago, and I'm really proud of the fact that many of these people we're still all working together. So Olivia Graham is now a PhD student, and Morgan Eisenlord in my lab. Maya was at one point a postdoc, and now she's a professor. And there's stories about all of these and how dedicated and brilliant they are, allowing us to do our work. This is our newer NSF group led by, here he is, Emma Duffy, and our postdoc Lillian Aoki. So I want to end on another note. Rachel Carson also loved creatures of the ocean, and this may be a side of Rachel Carson that some of you have spent a little less time with. She also wrote a book called The Edge of the Sea, and I want to end my talk with a tribute to our ocean biodiversity, kind of in her name. And these glass creatures are Blashka glass made 160 years ago by glassworkers that created over 800 of these exact matches. And my book here is about our quest to go back and find these living matches to kind of use it as a time capsule. But I want to say just a couple more things because there's a linkage here with Rachel Carson. And I really like this quote of hers that I ran across. Hanging from the cave roof were the pendant flowers of the hydroid tubularia, fringed and delicate as the wind flower. Here were creatures so exquisitely fashioned they seemed unreal, their beauty too fragile to exist in a world of crushing force. Yet every detail was functionally useful, every stalk and hydrant and petal-like tentacle, fashioned for dealing with the realities of existence. And this just amazes me because this is one of our Blashka glass models, also of the tubularia. Here's what the living one looks like. And it just reminds me that they both had the same vision of the exquisiteness of this amazing sea creature and how much they appreciated it. And so there are quite a few lines like that in Rachel Carson's books. And I have to conclude she's one of the greatest ocean writers of all time. And so I'm going to end there. And I'd be happy to take questions.
Okay. We live now? Okay. All right. Thank you, Drew. That was an amazing presentation, just very interesting and amazing. I'd like to encourage the audience, if you have questions, to please put them in the question and answer box. And while people are thinking about some questions, if I could take advantage of being a convener here and ask Drew one question, just get things started is, in your discussion of the sunflower seastar, you mentioned that it had been, a proposal had been put forward for listed as endangered. Has anything come from that? Do you have any update on that?
Oh, well, thank you for everybody for listening and for that wonderful question, Eileen. As it turns out, today, the IUCN went live with the listing the sunflower star as critically endangered. This is work that was championed by a number of people, the Nature Conservancy, the Seattle Aquarium, and led by Sarah Gravem and Sarah Hamilton. And it's gone live today. And so this species is endangered critically across its entire range with 97% reductions in the continental US and 90% across the entire range. And so this was one of the sea stars that was as common as a robin at the beginning of the outbreak in 2013. And so It's definitely, an impressive change through time, and we've been kind of watching it. And kind of, as I said in the talk, it was in our early experiments the most susceptible of the 20 or so species that were observed to be affected. And so, again, it seems to be the result of a multi-host pathogen um, driving, we hope not to extinction, but certainly to endangerment, a very common and ecologically important taxon.
Right, thank you. We have some questions now in the question and answer box. The first one here is from Phoenix Garcia Tigaros, I think. And the question is, how do these diseases spread among different ocean species?
Oh, that's a wide open question. But Phoenix, you've completely picked the big black box. Of course, understanding transmission is very poorly known within the ocean, and it's one of the reasons why we're starting to focus with the eelgrass in understanding how this pathogen transmits. And as I said, we were impressed when we discovered that the plants 400 meters outside our beds were picking up infections as at high a level as the ones inside, which means that this is waterborne and freely transmitting. Similarly, although we don't really know anything about the transmission of uh, the pathogen for the seastar wasting disease, it must surely have been waterborne because, again, captive populations within the Seattle Aquarium, the Monterey Bay Aquarium, and the Vancouver Aquarium were all affected at the beginning of that outbreak. So I think, for different pathogenic agents, different hosts, the modes of transmission will vary.
Okay, thanks. All right, we have a question here from Susan Roberts, and she starts out by saying, wonderful talk. And then she asks, is the relationship of R naught with higher temperature a consequence of increased susceptibility of the host species or increased abundance of the pathogen or other factor?
Oh, nice to hear from you, Susan. Thanks. Thank you for that great question. Well, there's a lot of work that could be done to try to unravel how the different causes of R naught. I think it's all of those, and it varies in different pathosystems. So, for example, we know for corals, they're very close to their upper thermal tolerance, and so the host is stressed, which makes them more susceptible to disease. So in that case, it's the host susceptibility that could be affecting the R naught. We also know in cases where we're able to culture a pathogen, so for example, with our eelgrass protist, that it becomes more infectious at higher temperatures and it actually grows at a faster rate. So we might say that the virulence in that case of the pathogen increases, or the R naught increases with temperature. So really important to figure out these dose response relationships. And I think, we're dealing with our own pandemic right now and coming to terms with understanding dose response relationships, and in the context of the COVID epidemic, that different environmental temperatures, different seasons, different levels of humidity and temperature affect also the risk of our getting an infection.
Okay, thank you. Another question here from Dwight Owens. With eelgrass disease, can we conclude that cold winter water temperatures reduce abundance of the pathogen?
Oh, thank you for that nice question. of course, we initially were focusing on summer temperatures, and so it was really exciting to see that we also needed to be thinking farther back about the temperatures in a previous winter and the concept that there might be reservoir populations of a pathogen that were affected by winter cooling and warming. And so that will definitely be on our list of things to investigate, is how those winter temperatures are actually affecting reservoir populations of the pathogen. But yes, as you ask, it seems likely that those colder temperatures are inhibiting, killing, or preventing overwintering of the pathogen. And this is something that's long been known for terrestrial crop diseases. There are pretty interesting forecasting models that have been developed for terrestrial crop diseases, and they include winter warming and cooling. And so this is actually a little bit novel in the oceans, but a kind of a sensible result.
Right, thank you. Another question from Linda Bradley. Okay, she says, given the variation in eelgrass wasting disease across the eastern Pacific, do you think temperature is the main driver of the disease outbreaks? And then she says, I asked just because I would have expected that those sites are in close enough proximity that temperature would be fairly similar. Do some sites have other important components to why their outbreaks are worse? And then she says, super cool stuff.
Thank you for the kind words, Linda. So I didn't have a lot of time to go into the complexity of all the factors that might be affecting risk of disease. We think temperature is a big one, but also salinity is very important. Probably eutrophication and siltation and other factors are likely to be important. So I don't want to oversimplify this by implying that it's only temperature is the driver. Interestingly, your point about temperatures being similar within a region, that's actually not the case. We're finding that we get a fair amount of variability even within the San Juan Islands that affects susceptibility of our plants or their risk of infection. And the data that I showed across the wide latitudinal range from San Diego to Alaska, again, is interesting. Of course, the absolute temperature varies, but the driver that was significant was the temperature anomaly at each site. So it was sort of how much of a change there was locally was more important, suggesting again that those plants and perhaps the pathogen are adapted to a particular regional temperature, and when that changes quickly, it can affect the host pathogen interaction.
Right, thank you. There is another question here from Dwight Owens. Why were there fewer infections in Oregon compared to Washington and Bodega Bay? And that was probably in relation maybe to the seastar.
I think that's probably in relation to the eelgrass, because Oregon really stood out in that latitudinal study that I showed. And in fact, it was really a problematic site the first year because the Oregon researchers had so much trouble finding lesions, and the levels were really low. And so it was pretty exciting when Lillian Aoki did the temperature anomaly analysis and showed that, if anything, those Oregon sites were anomalously cold that year. So the Oregon site, in some ways, was the site that kind of proved that relationship. Although, again, this was our first year. I want to be careful about making conclusions. But we do think that a big factor at the Oregon sites is temperature, but we're certainly going to be investigating this over the next 3 years, and trying to figure out what other factors might be important.
All right, just looking here and see if there are any other questions.
Submitted one.
I'm sorry, what?
I just submitted one. You might see it.
See it hopefully here in a moment.
Yeah, this will be a
one. All right. Maybe while we're waiting for Claire's question to come through, I'm just going to ask, with your eelgrass wasting disease pathogen, are you able to separate out the pathogenic and apathogenic strains? Is that a possibility?
Yeah, that's been one of the exciting new discoveries. Our colleague Colleen Burge and her students have been working on sequencing the genetics of the different strains. And my PhD student, Morgan Eisenlord, has also been isolating different strains. And so they performance tested 12 to 14 strains, and about half of them were apathogenic, which means that when inoculated onto the eelgrass blades, they didn't create lesions. And then the other half were highly pathogenic, so they rather rapidly created lesions and infections on the blade. And excitingly, in the initial sequencing is showing genetic differences, that the phylogeny they're falling out separately. So there's the possibility here that there's a genetic difference between these pathogenic and apathogenic strains of what are currently called Labyrinthial zosteri, but there may be some revisions needed in the taxonomy. So certainly a focus of some of our future work is to understand a little better, especially how those virulent pathogenic strains are circulating.
Okay, thank you. All right. You see if there are any other questions coming in.
And it's kind of just to add a little bit more to that, Eileen, because it is something we're really interested in. That Labyrinthial zosteri is kind of classically categorized as an opportunistic disease, that sometimes it's present but doesn't cause disease. And we're starting to think it may well be that we have a portfolio of different pathogenicity strains, and some of them are truly pathogenic. They're always a pathogen when they occur. But when you look across all of those strains together, those apathogenic lines cause the appearance of it being an opportunist. So there's some big mysteries to solve about the biology of this pathosystem.
Right. Okay. So following on that, there's a question from Cara St. Pierre. She's asked, is there any indication of a relationship between eelgrass wasting disease and freshwater inputs to the Northeast Pacific?
There's been some beautiful work in Sweden showing, so remember, this is a pathogen that's pan-global, that does occur in Europe and has been problematic in Europe as well as in the United States. And there's been some beautiful work in Sweden showing correlations between risk of infection and salinity. So low salinity conditions can inhibit the Labyrinthial zosteri. And we actually use this in the lab to knock back the possibility of any incoming infections as a pre-treatment in some of our experiments. We run them through a low salinity 10 parts per thousand rinse. The data, we do not currently have good data on the effects of salinity in the Pacific Northwest. So that's certainly one of the questions that we're interested in pursuing. And I don't have the answer on that because there's also the possibility that hypersaline conditions as well as low saline conditions are important.
Okay. So there's a question here from Mike McFadden, and going back to the seastar, he says, ask, what are the origins of the pathogen that has affected the sunflower seastar? Has it always been in the environment, but only active as ocean temperatures have risen? Or a mutation of an earlier, less lethal pathogen?
The identity, the origins, the current distribution of the infectious agent in the seastar wasting remain highly controversial. It's our early experiments working with Ian Hewson supported the role of a virus or a viral sized fraction as being important. There was a candidate pathogen suggested, adensovirus, but because there were no cell lines for echinoderms, that has never been able to be confirmed, the actual identity of the pathogenic virus. So I think there's a big mystery there, and it does point, as of course it won't be surprising to many of you, of the challenge of getting enough funding to do some of this work in such a quickly moving situation as a new disease outbreak. And so there's a lot of samples we wish we had from 2013 and 14 that might help us now in some of that evaluation. So we really don't know what the origins were of this one.
Okay, thank you. The question from Claire Reimers here is, she asks, is any evidence that coastal upwelling that brings cooler, nutrient-rich water to the West Coast influence disease levels?
Oh, I think so. Yeah, Claire, I think the upwelling can be really important. It was pretty interesting in the initial outbreak of the seastar wasting disease that disease levels were really low in Oregon initially in the winter when we were seeing outbreaks everywhere else. The seastars were doing just fine in Oregon, and we don't really know, but one possibility is that the upwelling in those cold waters were actually somewhat restorative or resisting the disease risk for the seastars. I think it'll be interesting with the eelgrass to be able to also look more closely at upwelling influences and how that's affecting some of our variation in temperatures seasonally in different locations. I don't really have an answer yet for that.
Thank you. A couple more questions here. This one is from Nature McGlynn, and she asked, what might be the best way to convey the importance of native coastal ecosystems in terms of disease fighting and so many other services to our local communities?
Thank you for that great question. I think the health link is very important. And so we also found this in locations we were working in Hawaii, that when people realized actually the level of pathogenic bacteria in the waters close to their homes, they were quite motivated to try to help with water quality solutions. And so that's one of the reasons we kind of started that project in Indonesia, to try to bring together in a kind of one health vision the notion that the health of our ecosystems and human health are tied. So I am hopeful that as we discover more about the links of natural ecosystems in basically pathogen cleansing or deactivating pathogenic bacteria, that this will help us increase the valuation and the conservation of these ecosystems. And certainly in the Pacific Northwest, these eelgrass beds are really good prospects for that kind of work, just because they're so well developed. They're occurring in almost exactly the places we need them, at least in a place like Seattle. That they're right sort of in ground zero for places where pathogens are actually being added into the water. There's a lot of mysteries, and we don't actually know what is the main sort of deactivating agent in some of these beds. We think that oxygen is very important. Certainly, that's the main component of sewage treatment plants. Is oxygenation, deactivating a lot of bacterial pathogens that require non-oxygenated conditions. But of course, the filter feeding for many of our biota in these eelgrass beds could also be pretty important, like as I showed in the oyster example. And then the final level that we would like to learn more about are the microbiomes, the actual bacteria living on the surface of the eelgrass, and their potential role in changing the environment for other pathogenic bacteria. So I think as we fill in some of these knowledge gaps, we'll be able, I hope, be able to communicate better to people the real value and the services provided by these nearshore ecosystems. And although we've been working on eelgrass beds, certainly kelp beds may provide some of these similar services.
Okay, thank you. Sort of following on with some of the eelgrass, Peter Goro asks, has the eelgrass disease made it to the Atlantic coast?
Oh yeah. So I didn't have time to talk about this, but in the 1930s and 40s, there was a major outbreak of what's thought in eelgrass beds that decimated large uh, large meadows and caused some ecosystem collapse in those locations. And that's thought to be, again, a similar cause, although the pathogen at the time wasn't isolated. So yeah, labyrinthial zosterae is very much present on the East Coast and does indeed cause a fair amount of destruction in those beds. So it is circumglobally distributed. Yeah, I can see there's a bit of a delay here. Sorry, I don't know why that is, but hopefully
Sorry,
I'm making sense to you.
it's working fine. A couple more questions came in. John Harding asks, he first says it's a very interesting talk, but he asks if you see any El Nino signal in the West Coast eelgrass outbreaks over time.
We haven't looked specifically just at El Nino, but I would think there would be. And this year we're very interested because we're into a La Nina year. We're hopeful that maybe this year will be a good year in the sense that we would predict lower levels of disease due to the higher rainfall expected and the lower temperatures expected in our Pacific Northwest beds. And so we're kind of looking forward to something more positive. And indeed, last summer, I think we even had the beginnings of some La Nina conditions with cooler temperatures, and some of our beds were looking better even last summer. So this will be an exciting chance to actually see we hope something get better. That's really our goal, is to figure out what are the conditions that would actually make some of these ecosystems do better.
Okay. So a couple last questions here is one from Dwight Owens, and asks, could humans or ships be vectors for introducing pathogens from different geographic regions?
Oh yeah, I think absolutely. And there's certainly records of, some of the most extremely well understood introductions have to do with aquaculture, and, for example, shellfish being moved around the globe and carrying pathogens with them. But there's certainly potential for, along with other invasive species, pathogens to be brought in shipping and other kinds of human transport. Yeah.
Okay. All right. And so from Jarrett Burns here, is there any indication that wasting or something similar has been present in New England? And many sites with historical high densities of seastars have few, if any, these days. And it's been so far for several years. So it's been that way for several years. So is seastar wasting a wider phenomenon?
Yeah. Hi, Jarrett. Thank you. Thank you for listening and for your question. And seastar wasting did seem to occur on the East Coast. In fact, there were reports of it in 2011, actually in advance of our big epidemic on the West Coast, which was 2013. There was some work also providing evidence for the role of a viral-sized fraction and those East Coast stars as well. So yeah, I do think they were quite heavily hit. And there were a lot of reports of wasting during the same period. I haven't seen more recent data about how those two species of Asterias may be recovering or not. I don't. That's unknown to me.
Okay. So Claire asked an interesting question here. She says, are there any long-term ecological research sites considering eelgrass beds on the west coast of the U.S.?
That's a good question. That's something we could look for. That's a really good idea. There are places like Morro Bay that were very extensive, large eelgrass beds that were drastically diminished over about a 10-year period. And so there certainly are quite a number of locations where there have been big declines on the West Coast. And some of that may be due to this disease, but that, it wasn't investigated at the time, which is why we're trying to do that work now. We are working in a really large bed, the Padilla Bay National Marine Estuary. It's reported to be the largest eelgrass meadow in the continental U.S., and so we have monitoring ongoing there, and there is in fact high levels of the wasting disease there. So we're hoping with some of our drone monitoring to be able to actually pick up changes in the aerial extent of these beds and develop methods that can be used sort of more widely for a larger monitoring program. Yeah.
Okay, thank you. I have one last question here, perhaps, is are there any mitigation measures that can be taken to deal with eelgrass wasting disease?
We're very interested in that, Eileen. Freshwater is one thing we're thinking about. There may be estuarine sites or places where water flow could be continued, and it's possible that increased low salinity water could be protective for some eelgrass meadows. It's not in general for marine diseases. We don't really have good control measures, right? On land, we quarantine, we vaccinate, or we cull. And none of those are very practical for most of our marine biota. And I sort of talk about this in the last chapter of my book. What can we do? But I think with the eelgrass, one thing that could be interesting is to try some controlled culling of infections. That would only work on a small scale, but it might give us some ideas for some larger scale approaches. The combination of maybe culling and controlled freshwater flows, and that could be really important. For example, we also have a project to try to plant back eelgrass beds. So we're working with the San Juan County in a restoration project. And that those mitigations could be very important when you're trying to establish a new bed. Is to, one, be sure you're not transplanting diseased plants, but also to find ways to give them a start but before the pathogen can take hold. Yeah, so that's a really interesting question we do want to pursue.
Okay, thank you. Great. Are there any, let's see if there are any other questions here. Look right. Right, okay, so Mike McFadden has submitted another question. All right, and he asked, let's see, there are a number of efforts to predict disease outbreaks affecting humans using as one input, environmental data. Are there any similar prediction efforts for marine diseases like you have described?
Yeah, actually, we were working with a worldwide group on coral diseases over a decade ago and trying to develop, because temperature is such an important driver of many of the, not only coral bleaching, but also infectious disease. There are forecasting models that have been developed by NOAA to predict particular kinds of marine diseases that are affecting the corals. So that's one area that's been pretty well developed, and it has grown out of the bleach watch programs that NOAA has developed. Coral bleaching is determined largely by temperature, and so forecasting works extremely well. And those algorithms can be modified now to try to account for some of the infectious diseases affecting corals. And that gives a chance for closures of public parks, for example, for corals, for scientists to be able to be on the spot when an outbreak is happening. Another place where that's being applied is again work that folks like Jeff Maynard and NOAA are doing with lobster shellfish disease. Again, that's one that is temperature sensitive, and there's a concern about the spreading of epizootic or the shell disease from Long Island up into Maine as waters warm in the Atlantic. And so there are quite a few places where applications of temperature-based surveillance and forecasting could be applied. And we're investigating this also with the eelgrass, whether once we refine the conditions with temperature, whether we can apply forecasting algorithms.
Okay, thank you. Right. Let's see. Don't see any additional questions here. I'll ask the audience if there are any other questions. Give people a moment for that. Very interesting and very thought-provoking presentation. So thank you for that. It's made people very aware of
Yeah.
the issues surrounding marine diseases. So thank you.
Oh, you're welcome.
Right.
Well, thank you very much, Eileen. And I really want to thank the group for your interest and your great questions. I wish we could all be together in one room, but it's pretty cool, at least that we can run this virtually. And I really am so grateful to AGU for their just phenomenal job of running this entire meeting that's been so exciting. And some of the other talks I've been to have been really wonderful. So this is a real tour de force, and I'm just very privileged to be part of it. So thank you.
Right. Thank you for your presentation. And we also should thank the AGU staff and support people from Entrado and everywhere for making this virtual meeting a reality. So thanks to them for doing that. All right. If there are no other questions, then I think we'll go ahead and say thank you and end the session. Claire, do you have anything you want to say before we do that?
Just a reminder to the audience that if you missed part of the presentation, or it will be available on demand, just go to the named lectures page and scroll down, you'll find it.
Okay, good. Thank you for that. Okay. So again, Drew, thank you very much for your presentation. Thank you. And I think we'll end the session at this point. So thank you all. Okay. Bye-bye.